Thank you for joining us today. My name is Justin Walsh, and I'm a covering healthcare analyst at Jones Trading. I'm here with Dr. Marc Hedrick, President and CEO of Plus Therapeutics, ticker PSTV. We're here to discuss current developments in Plus's pipeline and potential clinical direction. If the audience has any questions, you can submit them via the Q&A chat or send them to me via email at jwalsh@jonestrading.com. We'll do our best to get to your questions, time permitting. So let's jump right in here. Marc, thanks again for joining us.
Hi, Justin, thanks.
Before we discuss the specific clinical trial progress that Plus Therapeutics has made, why don't we start by addressing how you view the ultimate positioning of rhenium obisbemeda in the treatment landscape of CNS cancers?
Yeah, great and interesting question. So positioning is really important for us. And I think the best way to think about positioning is break it down in components. So let's talk about rhenium obisbemeda for a minute. So there are two parts of that. One is the isotope. So it's a unique radioisotope. It's not one of the more commonly used. I think it's an up and coming and very interesting radioisotope with the potential broad utilization. So the physics of rhenium, in our view, are ideal for the CNS. If you look at the energy output, you look at the fact that it's a diagnostic in the sense that the killing component is beta energy, the gamma particle for assessing where the drug is and absorbed dose has been incredibly valuable from a development perspective. We just allows us to really learn a lot from each patient because we know exactly how much absorbed dose is going to the tumor, and we know exactly where that dose is at any point in time through pretty straightforward SPECT CT imaging. Also, with respect to the isotope, I think one great thing about it is the duration of the radioisotope or the energy. So you've got about 90-hour half-life, which makes GMP centralized manufacturing doable. So we can batch manufacture this every week and send it out to places. And then the other thing about the isotope that I think is really important is the path length. So with the 186 radioisotope, the path length is 2 millimeters. So given how we deliver the drug to the CNS, that ends up being an ideal path length to kind of hit the tumor, but not hit normal tissue, and allows us to be very precise and targeted about where we put the drug. So that's the rhenium. Then the obisbemeda part is kind of a different element, and the positioning of that is kind of it intertwined with how we deliver the drug. So it's a liposomal formulation, and what that does fundamentally is it changes the pharmacokinetics of the drug. So for example, if you put the rhenium alone into the brain, it's gone within hours. If you, in the obisbemeda liposomal framework, if you put it into the brain, it's there for a few weeks until the isotope is fully burned out. So you essentially have no systemic absorption of the radiation, which takes bone marrow issues and renal and other toxicities just completely off the table. And that's frankly what we've seen in the data. So, and then also in the leptomeningeal space, another key potential opportunity for us commercially. You put the drug into the leptomeningeal space, which is the CSF, the cerebral spinal fluid space. Then you see it circulate very rapidly. So the liposome really kind of helps. So it doesn't discourage. It's really fast circulation throughout the CSF space, but you still see the drug there 7 days after a single administration. So that pharmacokinetic lengthening of the drug's dwell time or presence in the region of interest allows us to see what we think is meaningful clinical benefit with a single administration. And we think there's opportunity for multiple administrations, but even with single administration, we're seeing very promising results. And then Just very quickly, the third part of the positioning is delivery. So there's different ways to target radiation, for example. So there's external beam radiation, which is a beam, and there's a level of precision there, but you're getting a lot of normal tissue along with that beam, and you really have a limit in terms of how much radiation you can deliver. With targeted radiotherapeutics, so we think about molecularly targeted radiotherapeutics, you still have off-target effects. Radiation still goes elsewhere. So you're sort of fine-tuning how much can I get to the tumor versus, what is the critical organ toxicity level that I need to, make sure I don't hit. Whereas with us, because of CNS and unique anatomy and so forth, we use direct targeting. So for GBM, the ecosystem has developed such that you can precisely put a catheter within 1 millimeter of your intended place, or multiple catheters, put the drug right there, pull the catheters out, and that's it. There's no systemic toxicity, and there's essentially no toxicity or radiation delivery outside your region of interest. Incredibly important. And then also in the leptomeningeal space, that's where the tumor is. The tumor is in the fluid. You have circulating tumor cells in the fluid. You need to sterilize the fluid from those tumor cells, or the tumor is on the thin lining around the brain spinal cord called the leptomeninges, and it's rarely nodular. I mean, if it's nodular, it's not that deep. So if you get a 2 millimeter penetration, you can target the fluid column, you can target the leptomeninges, but then you don't get into the white matter and get into some of the complications you get with CSF delivery of methotrexate. You get white matter changes or leukoencephalomalacia, but you avoid that with this. So it's the isotope, it's the formulation, and it's the delivery. And I think that's where we want to really differentiate ourselves from everybody else that's going after these kinds of tumors with these kinds of drugs.
Got it. I think that you had some KOL calls that address this in depth, but maybe just quickly mention the fear. I think is always that you'll get toxicity or radiation dosage to the healthy brain tissue or the healthy CNS when you're delivering radiation directly there, and I guess the fear is that might be more of a concern when you aren't targeting a certain something that's molecularly targeted. So maybe you can just talk a little bit about maybe the measures that are in place to evaluate that, and just what you guys have seen overall, with the understanding that I think we've had, I think Dr. If I'm correct, Dr. Brenner talked extensively about that.
Yeah, it's really a good question, and it was a question that the FDA had expressed to us early on in the development for the GBM indication, for the leptomeningeal indication, and now as we're going back and forth with them with respect to pediatric brain cancer. So the part of, in looking at it from the FDA's perspective, they have concerns that are really tied up into external beam radiation, which is fundamentally different kind of radiation than nuclear radiation that you, with your background, Justin, know a lot more about than I do, but it's fundamentally different. But that's their bias coming into a meeting. So we really had to prove to them over the course of all of our clinical development programs that kind of florid neuronecrosis is not an issue. And that's why the dose escalation phase on the GBM with LM has been, in my view, painstakingly slow. And it's kind of, to me, I wish we could have moved a lot quicker. But so you kind of divide them up. And in terms of GBM, we're just not seeing the kind of neurotoxicity you might think. Part of it's just the precision of the delivery. But the level of imaging allows you to see, motor tracks, visual access, and so many kind of important functional tracks within the brain. You can really get precise about those things as well. But even, when we're, the disease mandates, we have to get very close to those. We're still not seeing the level of neurotoxicity you would think with delivering 10 to 20 times the amount of radiation you get by EBRT. Our hypothesis is that those normal brain cells are not, they're not mitotically active. So we know we're creating lots of double-stranded DNA breaks, which is why we're killing the cancer. These GBM is very highly proliferative, very aggressive, very, very infiltrative. So you seem to be clinically now in getting close to maybe 50 or 60 patients total treated in the different kinds of tumors. Just not seeing that. You're not seeing it in the imaging. You're not seeing it in the clinical findings in the patients. And we even had one patient that the doctor was concerned with some swelling in the brain after treatment. They went back and reoperated on the patient. And there was no tumor and no necrosis. So it's edema. And that edema is expected. If you're killing cancer cells, there's going to be a process to get rid of that the tumor that's dying, and so there's some edema related to that, but that's actually very treatable with steroids or with bevacizumab. So now we even have an example of a patient where we went back reoperated on it, didn't see the kind of necrosis that maybe was a concern early on. And kind of tie this back to our discussions with the FDA. We just sense the comfort with FDA each successive meeting, whether now we're talking about pediatrics or other indications. They're just a lot more confident and excited, frankly, about the technology, not only from now a safety perspective, but now we have efficacy signals that are really promising.
Got it. You just mentioned that the progress has been a little slower than you wanted, but obviously you're still moving forward. And just yesterday, you announced that the dosing was completed in cohort 5 of the phase 1 ReSPECT-LM trial of rhenium obisbemeda in leptomeningeal metastases. In your last call, I think you announced that interim safety and feasibility data from the trial is expected at SNO ASCO CNS conference in August of 2024, and potential for full phase 1 data at the SNO annual meeting in November of 2024. So maybe you could just start by reminding us what safety and efficacy data has been reported so far for this asset in LM.
Yeah, happy to. And the predicate for presenting it at those two meetings is really how we're thinking about the development in terms of where we think the maximum tolerated dose will be. And if it's anything like GBM, we haven't gotten to a maximum tolerated dose despite delivering, really unheard of, never performed doses of radiation to the brain. I don't think we'll get to a maximum tolerated dose in LM. To answer your question specifically, I think probably cohort 7 is going to be at the sort of at the end. I doubt we'll get to toxicity. If you look at the absorbed dose to the leptomeninges, the CSF, and compare that to the dose you get to the spleen. Splenic dose has been flat, well below critical organ level through all cohorts so far, through cohort 4. I can't speak to cohort 5. And you see a stepwise linear increase in the amount of radiation delivered to the neuroaxis. So I just don't think we're going to get there even at cohort 7, but practically we're sort of beginning to exceed. I mean, these are just unheard of dose levels, but we're sort of beginning to exceed our level of practically manufacturing the drug without much radiation. So, but also I think our plan is to look forward. We could talk more about this in a minute, getting into multiple doses. It just makes sense that we want to maximize the aggregate dose over time. To in the case of LM, we don't want patients dying of their LM. If they die of their primary disease, that's something we're not going to be able to control, but we want to take dying of your leptomeninges off the table and improve the symptoms dramatically in those patients. That's what we hope to do. So in terms of safety, we've treated now, I think, 18 patients in the LM trial. as I mentioned, critical organ dose toxicities are low. Therefore, it makes sense that we're not seeing systemic toxicities, which we're not. I mentioned a minute ago that the drug stays in the CSF space for at least 7 days, maybe much longer. We don't know. It's probably taken up by peritumoral macrophages, which tends to anchor it and make it more focused around the tumor, but also we know it continues to circulate as well. So, so from a safety perspective, we're not seeing any neural signs of toxicity or systemic toxicity at these levels, and I don't anticipate them, given kind of what we're seeing in terms of measured absorbed dose. Back to the fact that there's a gamma particle, we can measure it, we know exactly how much we're delivering, we know exactly where the dose is. It's really useful in kind of thinking about the development pathway, and it helps kind of project out where we're going to be. So we have certain rules with respect to how quickly we can enroll in our trial. So and we enroll once we have availability to enroll, we enroll, within the week. I mean, it's just critical. Really, it's critical to the timing of the development to be able to do that. And we've got great investigator interest in the trials. So I think we'll complete cohort 7. We'll do a data update as it relates to safety and at SNO ASCO, and likely have the full trial completed and report that data at the SNO meeting in November. Now we'll continue to follow up those patients. So we could talk about efficacy data if you'd like, but that has implications for how long we follow those patients.
Yeah. So what have we seen in terms of efficacy? How are you tracking that? And what do you, and tied into that, what do you think investors should be looking for in your updates to help convince them that the drug is providing a benefit? And I know we're dose escalation, and the focus is still largely on safety, but people want to get some sort of a glimpse in the efficacy in any way that they can.
Yeah. So efficacy, we're still early, and although we think about efficacy, it's hard to say something compelling about efficacy at this point. Although what we see is certainly, I would say, very interesting, if not, very promising. So if you think about it in terms of biologic response, one of the things we've looked at is tumor cell counts in the CSF. Thinking, okay, well, if we're reducing the number of tumor cell counts in the CSF, then that should be a sign of efficacy or be linked to efficacy. The problem is the assays for that have largely been unchanged since I was in medical school, going back many years. They're not sensitive, they're not specific, and it's more witchcraft than it is science, frankly. So we were very excited to work with a company that had developed a highly specific, highly sensitive, greater than 90% for both, assay for enumerating or measuring, quantitating tumor cells in the CSF using 18 different monoclonal antibodies over a whole panel of adenocarcinomas and melanomas, which are the core of these, the primaries here. So we looked at that, and we saw an average of 50% reduction in the quantitative tumor cell counts at 28 days after treatment. So you see kind of very early, you see a little bit of a decline, and then by 28 days, you see a pretty dramatic decline, up to 90% of in one case, I think 90% reduction tumor cell count with a single administration. And then you see it recovering back at 56 days. So to us, that's a very interesting sign. Now, that's not an approvable endpoint because that's exploratory endpoint, but from understanding the biology of the disease, that's very promising because you're clearing out the space where the tumor is circulating and sitting. So that's kind of one sign of efficacy. And also we've been following the patients in terms of their survival. We actually have a couple of patients who have survived long enough to come back and ask for an additional dose. So we've treated now a couple of patients under compassionate use. We have patients living out beyond a year. And Justin, we've talked about this before, and people listening may know, but the median overall survival for these patients without treatments is 4 to 6 weeks. in just a few months for patients that have breast cancer and lung cancer, which are the two most common primary causes of leptomeningeal disease. So we're seeing, I think we reported in November of last year, in the first 10 patients, a median overall survival of 10 months. That's with half the patients still alive. And we're seeing those patients just continue to do well. So I see there is an interesting efficacy signal there, and arguably more than interesting. But we're continuing to follow that, and we're very excited about multiple doses. One of the concerns the FDA had was, what's the radiation toxicity of multiple doses? Now we've got a couple of patients under our belts, treating them with very high doses, and they seem to be doing very well. So those two things, I think, are very promising in terms of overall efficacy. One footnote is the cell enumeration test actually went away for the latter part of 2023, and I'm happy to report that we've acquired that technology, we've implemented it, and in finishing cohort 5 that you mentioned we just reported, we've re-implemented that tumor cell enumeration testing, and our plan is to really ramp that up. We think that's a fantastic test. It solves an enormous problem in patients with CSF tumors, not only diagnosing them accurately with high specificity, high sensitivity, and ruling out patients that have maybe high risk of getting LM, but it's nice to be able to tell somebody with triple negative breast cancer that has neurologic symptoms, hey, don't worry, we've done 2 successive tests that show you don't have tumor cells that are there. And then finally, to follow these patients. And we inherited a data set that shows that you can actually treat these patients down to a 0 tumor cell count, follow them, when it comes back, retreat them. And so you can use those longitudinally to follow patients. So we think that assay has enormous potential impact to these patients. And then finally, just in terms of that test, we think that LM is about 2 to 4 times underdiagnosed. So if you look at autopsy studies, there are a lot of people that die with LM that's never diagnosed, and may actually double or even further increase the amount of addressable market. So having a highly sensitive specific test could increase the market size for LM and increase the commercial opportunity.
Got it. So maybe we can, you'd mentioned breast cancer. I think you might have answered this because it's the largest indication there, but why are you focusing on LM from breast cancer first? And do you have the expectations about what you might prioritize next?
Yeah, so I think part of what we're doing in the initial phase 1 trial is concentrating our treatment on patients with lung cancer and breast cancer. They just, if you look at the numbers, they're just the biggest opportunities. In other words, they're the places where there's the most clinical need in these patients. So we actually have we have orphan designation for breast cancer. We will get orphan designation ultimately, I believe, for lung cancer. But the FDA has been very clear that if we want approval for LM, we have to go primary by primary. So the development concept here is to work out our dose in a basket trial, get the maximum tolerated dose or maximum feasible dose, then work out what is the appropriate dosing regime over time that's safe for patients, and then get that approved. And we think a phase 2-3 pivotal trial is something the FDA will potentially go for, because there really is nothing approved for these patients. So essentially a single trial, perhaps with a lead-in, just to get some comfort in terms of the primary endpoint selection. So breast, it's the patients with breast cancer, it's the largest market. It's the logical place to go first. We have data that supports the safety and potential efficacy for patients with breast cancer, but I anticipate lung cancer will be right on the heels of that, and then also melanoma. Those are kind of our three top target areas, and then represent kind of a significant component of the overall LM disease opportunity.
Got it. You've, I think, talked about a number of your FDA interactions. I'm wondering what you can say overall about how your interactions have been related to rhenium obisbemeda in LM, as well as just your sort of plans and strategies around continued interactions with the FDA in terms of what you can actually disclose, of course.
Sure. So I've alluded to that a bit, but I'll provide a bit more color. So we inherited the technology with a single long-lived academic investigator-initiated trial for GBM. It was dose escalation, and so our first interactions with the FDA as a company, we had some data that showed safety. So, but they were still skeptical because the mindset really is around external beam radiation, not nuclear radiation, and nobody had started to dial up the radiation intensity like we had before. So I think there was just some trepidation about getting up to some of these higher doses. For example, if you look at recurrent GBM, if those patients get radiation, they get about 35 gray of radiation absorbed dose to the tumor. And we're getting, I think in our phase 2 trial, we're over 300 gray as an average absorbed dose to the tumor. So we're very high doses that they haven't seen before. So what maybe provide a little bit of color. I mean, what we're seeing from FDA is, I think, increasing comfort of the safety profile. So that's really helpful. In LM, for example, some of these patients have had external beam radiation, and so they've wanted us to exclude those patients from the trial initially. I think we'll be able to go back and add back patients that have had external beam radiation and then treat them with with rhenium obisbemeda for their LM complications. I think the FDA will allow us to move forward with that, given the safety profile that we've shown. Furthermore, I think we're very likely at a single dose administration at LM that is higher than what we will give with a multiple dose approach. But now we've got a buffer that I think we can go back and address those multiple dose concerns that the FDA had initially by actually dialing back the dose a bit, because we're seeing tumor cell effect at the lowest doses. So even at the lowest single dose levels of rhenium obisbemeda in the leptomeningeal space, you're seeing a meaningful improvement in tumor cell count. So I think we can back off that level, and it gives us some buffer with the FDA to set up an approach to derive a multiple dose regime to accomplish some of the clinical and commercial objectives that I mentioned before. So Dr. LaFrance, who's our chief medical officer, is, as he says, is in constant communication with the FDA. So that's sort of his style. We are talking to the FDA about our adult GBM program, our LM program, and then they've been a little bit, which is standard for them. They've been more cautious about introducing this into pediatric patients. And you've got developing brains, and you've got a whole different set of concerns that you have with an adult patient that's got GBM. So they've actually signed off on our clinical protocol with respect to pediatric brain cancer development. They just wanted to see additional dosimetry data from our adult trial before they allow us to move forward. I think we're optimistic that we'll get there this year and we can begin that trial. But that's really that's sort of the ultimate concept of being successful with the FDA. When they allow you to treat pediatric patients with super high doses, or treat adult patients with multiple doses that are also very high, that you kind of arrive from a safety perspective. And I think we're just about there with them from a safety perspective. And then it's really a matter of getting the trial design down for approval. And that's something that's very much front of mind right now.
Got it. And understanding that the trial design is still under consideration, and you're still enrolling patients and having conversations with the FDA. What is just your current thinking with respect to possible timelines to either initiate the pivotal trial, and then once initiated, what do you think would be a reasonable guess about the length of the trial? I know obviously if it's the trial design have a big impact there, but
Yeah,
what are you thinking right now?
yeah, I think that so. You have to divide, just for example, glioblastoma and
Yep.
leptomeningeal cancer into two separate buckets, because even though they're both brain cancers, they're just very different from a clinical development perspective. So because of the bevacizumab issue that the FDA had way in the past, and for people that don't know, bevacizumab is the only drug that's been approved for recurrent GBM in over a decade. It doesn't really prolong survival, but it was not approved on a survival endpoint, but on other endpoints. So they're very much wanting to see overall survival data for GBM, and that's probably the right answer. We've gotten their agreement that that's the right endpoint, and that's how we're thinking in terms of that trial design, in terms of going after overall survival. However, we will go after PFS and novel imaging endpoints. We're looking at kind of 4 different levels of imaging and on a patient-by-patient basis in our GBM trial. And we think we have some interesting findings there that will help support that, but ultimately it's going to be about overall survival. I think the key question from a design perspective is, to what degree can we use real-world data in that GBM trial? And Dr. Califf has recently the FDA has had a recent panel on using real world data. We've had multiple discussions with the FDA about using real world data. We've actually done 2 real world data trials as controls in our pilot GBM trial with our partner Metadata. And both of those trials that we've performed have shown about an 8-month overall survival for recurrent GBM, which is consistent with meta-analysis that have been published as well. So we think that 8-month standard of care is sort of the clinical hurdle rate as it relates to overall survival endpoint and powering out a trial. So we also plan in the phase 2 trial to do another real world trial as a lead-in to our discussions with the definitive discussions with the FDA about pivotal trial design. So we'll go in with additional real-world data. Now, the FDA has previously green-lighted a phase 3 trial design in GBM using real-world data. So we think, and that's our collaborator contractor in this, Metadata, was responsible for that with that sponsor. So I think we're cautiously optimistic that will allow us a real achievable path with the FDA to incorporate real world data. What that does for us is a couple of things in terms of the pivotal trial design. It changes the randomization scheme. For example, rather than having to randomize 1-to-1 to our rhenium obisbemeda to the comparator, we can actually randomize 3-to-1. So 3 out of every 4 patients, for example, would get rhenium obisbemeda, and only 1 patient would get randomized to the comparator. So that really helps from a patient psychology perspective. Patients with if you look at the published algorithm for how doctors are to treat patients with recurrent GBM, right up front it says you really should consider a clinical trial for all patients that have recurrent GBM. So clinical trial is, in many ways, the first option for these patients. So being able to randomize 3-to-1 to the active arm is psychologically really important in getting patients to participate in the trial. And then the plan would be to supplement that with additional control patients that that come from a real world experience database that are propensity matched exactly to the trial. And we think there's a high likelihood that will happen. So back to your final question about timing. I think that if we're able to do that, it reduces the number of patients that we have to actively enroll or actively randomize, which shortens the time frame. So, with 20 sites, you could enroll that trial at the likely active control patients in maybe a year, and then figure 6 to 12 months of follow-up. And our goal will be to get that started after we finish the phase 1, phase 2 data set, and then go to the FDA and get alignment around that. So it'll be a couple of years to do that trial and report that data. Not that far away. I think we're on track to get our phase 2 enrolled by the end of this year, maybe early next year. We're adding a number of new sites. As you know, that's funded by the National Cancer Institute. So there's certain site limitations that we have to work with in terms of the phase 2, but we're basically doubling or tripling the sites this year. So that impact will be at the end of the year, which is harder to forecast, but end of this year, early next year. So it's a long answer, but that's a very complex clinical trial plan, but it's very much front of mind. With respect to LM, we're probably on a similar time frame because I think the likelihood of being able to do a phase 2, 3 pivotal design is much higher. We're not excluding that with GBM, but I think because of the bevacizumab issue and other things, it's less likely. But very much, I think that's possible with LM. So we think that we'll be completed with our monodose single dose administration trial after cohort 7, which should line out around Q3, early Q4. And even prior to that, we'll be going back to the FDA. As I mentioned, Dr. LaFrance talking to the FDA fairly continually. And our plan is to take a single administration dose into a phase 2, 3 pivotal design. The primary endpoints will, in contrast to GBM, will not be overall survival. And the problem with overall survival in leptomeningeal cancer is that, in a way, the patients have two cancers. They have their primary cancer, whether it's breast or lung, and then there's the LM. And so they can succumb to either. So it makes an LM OS trial very complex. So we're talking to KOLs at major cancer centers around the U.S. who are experts in this, that will be with us as we go talk to the FDA to discuss our plan for clinical development. But it likely will be symptomatic improvement or some composite endpoint related to perhaps imaging, perhaps CSF measurements of cell count, even though it's not great, it could be incorporated in a composite endpoint. So it'll be an endpoint that will be less rigorous in a way than OS, but there isn't a competitor really. There's really nothing out there that competes, that sort of creates the hurdle rate there. So so in terms of timing for that, with that as the context, finishing up that sort of phase 1 dose escalation with a single administration towards the end of this year, getting hopefully agreement with the FDA about what a 2-3 design would look like. That probably incorporates sort of a roll-in or sort of a lead-in design to help confirm your statistical analysis. But then that can enroll in about a year. That will enroll very quickly. So if you just think about the epidemiology, in the US, there are 15,000 GBM patients newly diagnosed a year, and it's 10 times that for LM. And the number of competitive trials is much less in LM versus GBM. So in theory, I think everything goes well, that gets started up hopefully relatively early next year. That can be enrolled in a year and potentially be a pivotal trial. And just as a caveat, we do have CPRIT funding through phase 2, so that almost $18 million grant we have through CPRIT covers the phase 1 and the phase 2. And so potentially that would cover a significant amount of that trial.
Got it. You'd mentioned, so if we turn back to GBM quickly, you'd mentioned the impressive overall survival results. that were reported compared to the historical results.
Mm-hmm.
I'm just wondering if there's anything that you can say about what additional results we should expect in the second half of 2024, and what sort of indications of clinical benefit should we be looking at for the current RESPECT GBM trial?
Yeah. So as mentioned, we're looking primarily at overall survival, and so we presented an update on the overall survival in November, and that was median overall survival was 13 months with a number of patients still alive. So that's about a 60% improvement over standard of care, which is about 8 months. That is consistent with what we saw in the phase 1, although the phase 1 was dose escalation, and there were some patients in there, although it was a heavily pretreated population of patients. There were a couple of patients that didn't satisfy the 2022 WHO criteria for GBM. So they had less than GBM, and a couple of them kind of pushed that survival number out a bit more. But that 13 is pretty consistent with what we see in the phase 1. And that's one of the beauties of being in the radiotherapeutic space. If something works in phase 1, it's very likely going to work in later stage trials. And we're kind of seeing evidence of that. So OS is really important. I think the safety signals are such that I'm not anticipating any ongoing concerns about safety. Don't anticipate anything new there. There's just no systemic toxicity that we're seeing whatsoever. I think what's really potentially interesting to look at is the imaging endpoint. So we're looking not only at standard T1, T2 MRI images and follow-up, but we're looking at trams, at relative cerebral blood flow, and a new technique called flip books. And so this is where you start thinking about this GBM technology not as just another potential therapeutic for GBM. This is a new platform to deliver radiation to the CNS. I fundamentally believe that. So if you think about how new technologies are developed across all industries, high tech, biotech, whatever, you sort of see a new entrant come in, and then it gets improved over time, and then it kind of plateaus, and then you don't have a lot of ability to incrementally improve that going forward. External beam radiation for patients with brain cancer is there. It's been improved. It's sort of maxed out in terms of its clinical benefit. By personalized treatment planning, which is what we use a software called BrainLab. We can very specifically plan each patient and their delivery in a very seamless, cost-effective way. We customize the catheter placement, the number of catheters, the depth, and so forth, and the anatomy in that treatment plan. The catheter delivery is very safe. We've got extensive amount of experience with convection-enhanced delivery. Very safe, very predictable way to deliver radiotherapeutic to a tumor. And then you have the dosimetry and this follow-up imaging that allows us not only to deliver these super high doses of radiation, bypass the blood-brain barrier, bypass chemoresistance, work around complex CNS anatomy, 10 to 20 times the amount of radiation, and not only treat the tumor, but treat that rim around the tumor called the infiltrative margin. And that's really the problem in GBM. When you go take out surgically a GBM tumor, you almost always leave some tumor behind, and you never address the infiltrative margin, which is at 2 centimeters around the tumor, which is where all the recurrences show up. So our platform allows us to go back and potentially retreat patients that are inadequately treated, or ideally treat that whole infiltrative margin that you can't really treat well with any other therapy. So this is a platform that addresses all of the challenges that physicians see as it relates to GBM. So So I think this is a step function in terms of delivering radiation to the CNS. And I think we're just scratching the surface of where this could go.
Got it. Well, talking about stepwise technological improvements, maybe you can address the current status of RNL-BAM and some of the implications of that asset being deemed a medical device.
Yeah. I'll back up a bit. So we a little over a year ago, maybe it's getting close to a year and a half ago or so, we licensed a radioembolic drug at the time that we thought was a drug, which addresses some significant weaknesses of the two existing products that are in the market. And those products are permanent radioembolic drugs. They use yttrium-90, for example, but you put them into the vasculature, they deliver their radiation, and they stay there forever. They're glass beads or polymeric beads. Boston Scientific has one of those products on the market, and those are used principally for inoperable liver cancer. So working with academics who had developed a interesting design that we licensed for developing a bioresorbable drug. So the idea here is that with these non-bioresorbable drugs, you're kind of one and done. And you don't cure these. It's really about palliation and trying to improve survival and symptoms. But if you can tune your bioresorbable drug so that you deliver the radioembolic, it delivers its radiation, then it biodegrades, it goes away safely. There's no systemic toxicity, but it's not like you can go right back in and retreat if you missed a spot or it comes back. So that's an example of a differentiated property that what's going to be dialed into this drug, as well as better localization of that drug, dosimetry. We can use 186 or 188 radioisotopes, so it's versatile in that sense. So we not only think this could be a new entrant into billion plus market for treating liver cancer, inoperable liver cancer, but effectively you could treat any tumor in the body that you can access angiographically. For example, GBM. So what if you treat GBM by convection enhanced delivery, and it comes back in a space that's very difficult to get because of its proximity to a ventricle. You really can't convect into that because it's a low pressure sink. You could get there through the vascular system and deploy your radioembolic just upstream of that tumor, hit it with radiation, come out, single dose, single administration, no surgical procedure, minimally invasive surgery procedure, and allow you to continually come back and try to gain survival benefit for that patient. So GBM, pancreatic cancer, there are just a number of cancers that are really tough, but are very amenable to delivery through radioembolic. So we have really high hopes for that drug. But now it's not a drug because it's actually a device. So we went to the FDA and asked for requests for designation using the process they use now. And they said that this is more likely will be a device, not a drug. And why that's important is the quality system and the development work is different pretty substantially between a drug and a device. But from a development timeline cost perspective, where the rubber really hits the road is that it's a pilot pivotal to approval as a device, not phase 1, phase 2, phase 3. So we think it speeds up the development, actually makes it probably less expensive. It's a bit different quality system, but our team knows we're putting that in place right now. So right now we're trying to finalize the design of that drug so that we have exactly what we want when we complete large animal preclinical testing and get that into the clinic. We'll start out in liver. That's an existing market. There are existing reimbursement pathways. There are existing partnering candidates in that space that we're well aware of. So we think that's the and the liver itself is a giant organ. It's very forgiving. You don't have to be super precise because you're worried about complex neuroanatomy, for example. But it's a great place to start, and then that'll be a jumping off point for some of these more difficult cancers, like brain cancers, spinal cord cancers, pancreatic cancers, and others.
Got it. Now, maybe can you provide some additional color on your efforts to improve your supply chain and highlight how much rhenium-186 you expect you'll need to support pivotal trials and potential commercialization?
Yeah, Justin, you cover a lot of radiotherapeutic companies, and this is a question that comes up all the time. It's not, it's not your mother's supply chain in radiotherapeutics. It's often more complex, and you've got a radioisotope that has a shelf life of hours or weeks if you're lucky. So I think that the things I want people that are interested in the company, investors, to know is that we're right where we need to be at this level of development. So we have more capacity than we have patients who need drugs right now. So from where we are today in early 2024, into 2026, that plan is in place to ramp the two key components of the supply chain. The first is GMP manufacturing. So we don't manufacture the drug; it's manufactured through our GMP contractor. So we have enough capacity right now to manufacture about 4 doses per week. So that's enough to cover our current demand. In 2024, that will go up to 10 doses a week, and 2025 up to 20 doses a week. So that gets us through all conceivable clinical trial development scenarios. So that's kind of point number one. So that solves the GMP manufacturing. So, and it's really a forward-looking solution because we're where we need to be today. The other issue is radiation services. So we use MIR. We have about 4 times the capacity that we need right now in terms of radiation services to take the target and turn it into rhenium-186. And we anticipate through 2026, we'll have 3 to 4 times over capacity in terms of radiation services till then. So back to my original point, we're kind of where we need to be right now. But we're thinking about commercial launch in 2027. And our timelines sort of show that if things go well. So we need to augment both GMP manufacturing and radiation services. So we're working on that. 2024 is a critical year. We'll be adding a second GMP manufacturer. We're talking to a number of parties right now and going through that process. And then we're also talking to parties about additional radiation services. So our goal will be, once we get commercial, to have a primary supplier and a backup supplier, so that we can solve for any potential demand scenarios. GBM is the easier one because we anticipate that's a total addressable market in the US of about 9,500 patients per year. LM for breast, you start looking at 6 figures, maybe 100,000 patients per year. So that's where I think we need to solve for that fundamentally. But we're right on track in terms of where we need to be. And then it is a bit of a complex manufacturing process, but we've got it dialed in. And in terms of mitigating some of the intermediates, we just mitigate that through increased inventory. And the chelator is critical. We have a 10-year supply agreement that is exclusive to us. So we try to put things in place that not only give us supply chain visibility, but also barriers to entry for other companies.
Got it. So my last question here, wonder if we can talk about the current cash position, runway, and the role of awarded grants, and then sort of building on that. I'm just wondering how you feel if you've gained useful experience that could increase your chance of being awarded further grants, unlocking more non-dilutive funding. Obviously, with the understanding that there's never a guarantee that that'll happen.
Yeah. So in terms of cash, we reported just under $9 million in cash at the end of the year. We've been running, I think, since 2019, somewhere between maybe 12 and 20 million, and kind of cash balance. It's sort of been the range. We're a bit lower now, but we've offset that because we have grant revenue. So if you add, forecasted, call it 6 to 7 million in grant revenue in 2024, we're kind of still within that range of where we've been in terms of operating runway. So kind of to rephrase that from cash to operating runway, we've got cash to get us through early to mid 2025, depending on different operating assumptions. A lot of the investment for supply chain, for example, and developing GMP rhenium obisbemeda supply, that was done in prior years. So that investment is gone. We continue to benefit from grant support, as you mentioned. We still benefit from an NCI grant that is in place to fund the a lion's share of the GBM development program through phase 2. Thereafter, we'll have to fund that through a different mechanism, and we're putting plans in place to do that. In terms of our LM development, that's almost fully funded by the state of Texas. So state of Texas is the second largest funder of cancer research in the world. So they fund 2/3 of that. We fund 1/3. And we have funding, that's about an $18 million grant. We have funding for another year and a half or so, and that funds us potentially, as I mentioned, through phase 2, 3 pivotal. So we've had very good luck with grants, and we've got a team that's very accomplished in terms of getting grants. My olden days, I was able to get a fair amount of grant funding through the NIH and other places. So that's part of our strategy. It's part of our strategy to minimize dilution, minimize our trips out to the capital markets, to magnify the development pipeline more than we otherwise could with available cash. So our approach from a communications perspective is we won't talk about grants until we get them, because they can be really hard to get if you just look at the percentages. But last year we submitted 7 million in grants, and we're still waiting to hear back from the last of those grants that we've submitted. So we're still waiting to hear from those. We intend to kick that up to $10+ million this year. And so a lot of grant funding is just the reps. You've got to get grants in front of people, and they give you feedback, and then you work with them, and then one day hopefully you have funding. Just as a final caveat, CPRIT, it's a fantastic program. I think they're really supportive of Plus, of the LM indication, and of the technology. We're a Texas-based company. We'd have companies that have 3 grants. They will fund through phase 2 up to $20 million. So we think there's some opportunity there. They have 2 funding cycles a year. So we're going to continue to go up to the plate and take our swings, not only with the NIH, but other funding sources, and of course CPRIT.
Great. Well, we certainly look forward to hearing more about that and how it develops. It looks like we're just about up against time, so I'd like to thank Dr. Hedrick again for taking the time to speak with us today, and to our audience for dialing in. So have a great day, everyone.
Thank you, Justin. Take care.